So this lecture has a strange title. Let me tell you the history of this. We have at Weizmann a monthly lecture where every month another senior biologist is given a chance to talk about his work. And I was very surprised to be invited to do this. So I was thinking, what can I tell them? And usually what happens in these talks is that the biologist comes and in 1 hour summarizes his life. And so my life is not relevant for them, most of it, because most of it was spent in physics. So I said, okay, this would be a good thing to try to explain to them the kind of joy and difficulty that someone like me, who switched into biology at the age of 50-some, experiences. So I'm not sure whether it's relevant for you because all of you are young, but I hope it's not going to be boring. So I tried to make a list of physics versus biology as I saw it or see it. Again, upsides. I don't want to offend anyone, and I definitely do not want to spit into the well from which I was drinking most of my adult life. So what I did, I list here only the upsides, where biology is in yellow and physics is in green. And the downsides are by implication, okay? But I will mention them as we go. So first of all, in biology, what really attracted me to biology was the immediacy of phenomena. As you will see from some of the things that I'm going to tell you about, you walk into someone's lab, you look into a microscope, and you see something absolutely fascinating and ununderstood. You don't have to build a 27-kilometer long tunnel 100 meters under the surface of the Earth and have 3,000 people working on it for 7 years in order to see something interesting. And these phenomena are interesting and exciting in a way that can be easily understood. So this immediacy phenomena is something I miss in physics, in today's physics. Now in physics, there is the beauty of reductionism. Everything stems from some simple basic equations that are cast in mathematical language, and there is a perfectly respectable discipline called theoretical physics. And I'll stop here. In biology, right now there is an amazing number of really basic things that are not understood. And there is an enormous number of new technologies that are popping up essentially every day. They are measuring things today and are doing all kinds of microscopic manipulations today that 10 years ago would have sounded science fiction. You can go to take a living animal and input light, shine light onto its brain, and activate one particular cell that they want, or one particular ion channel in a set of cells that they want. Turn it on and off. This kind of manipulation, single molecule, single cell manipulation, no one dreamt about 10, 12 years ago, 5 years ago. And when you go to a biology conference, there is a smell of breakthrough in the air. I see people, many young people, they're all excited. Everyone is excited. I'll stop here about physics. And now in physics, this is an upside again. Many papers are what Pauli said that about a paper. This paper is not even wrong. So in physics, there are many such papers that are not even wrong. But there is an objective truth. So if you write you publish a wrong paper in physics, two things can happen. Either it is not noticed and ignored, or someone reads it, figures out that it's wrong, writes a paper explaining that it's wrong, and the minute his paper is published, your work is dead. It's immediate death. Now, that is not the case in biology. Nonsense has a long lifetime. And there is no objective truth. Biologists accept right and wrong by consensus. If there is 60 or 70% of the community believe that something is wrong, then it's wrong. Upside of biology, that one is frequently wildly surprised. So the fact that I'm surprised can be due to large extent to my ignorance, because I never learned biology properly. So maybe I would say people who know more are surprised less. But for physicists who go into biology, I promise you that you will be surprised right and left. And in physics, this is again an upside, submitted papers are reviewed. They are reviewed. That's point number one. And the reviews are very rarely completely idiotic and unfair. Whereas in biology, it's different. And this final point, if I get there, the last topic I will talk about covers this business of selection versus causality, which has to do not with upsides or downsides, but rather with our education as a physicist. Okay. So now this was the introduction, and let me give you the outline. I'll tell you 3.5 stories. The first one is a study we did on growth and differentiation in heart development. The subject is development. The second one is dynamics of transcriptional response of cells to stimulus. And the third one is what I told you yesterday, this breast cancer stuff. And I'll try to fill in and talk about things. Some of you will be bored, and I'll tell you when to wake up. Okay? Because some of the things you saw yesterday, but then I'll go and complete what I didn't show you yesterday. And the last one, this half, is about acute lymphoblastic leukemia in children with Down syndrome, and this will be my demonstration of causality versus selection. So, the first story, growth and differentiation in heart development. These are the actors in this story, and this guy is a PI in development, and she was his PhD student. This lady came as a postdoc at some later stage. He is my PhD student who got along extremely well with her, and that's why the project succeeded. These projects, collaborative projects, typically succeed when someone from my group has a rapport with the person from the other lab. So this worked extremely well. And so let me show you what happens when a chick embryo. Okay, in this lab they study the embryo of chicken. So they break a lot of eggs in this lab. And so this is now a movie of the second day after the egg is laid of the embryo. It's about 24 hours long. And just watch this. So these are called somata that are coming up. And at some point, the heart is formed. You won't see when. And then it starts beating, and then you'll see it. There it goes. Now, isn't this a miracle? So here is a beating heart. First, it starts with a tube. It's called the looping heart. Well, it starts with the stages before that, and then it starts to beat. And this happens after the end of the beating starts about 10 hours or 12 hours into the second day. So this is the, oops, I didn't want to do that. And within this big picture, we focus on a smaller one. After the heart started to be there and to even beat, cells from something called, from a region called the secondary or anterior heart field, which can be marked so you can identify them. They migrate towards the heart. As they migrate, they differentiate. So these cells are what is called early progenitor cells, which means that if given the right signal, they can become one of many different things. And they are given the right signal on this trajectory, signal being one of those chemokines that Antonio was talking about. One of those morphogens, molecules that is a receptor that catches them and then causes something. So they wander down, they reach the heart, they join the heart as cardiomyocytes, as fully differentiated heart muscle cells. And then they form the upper right ventricle or something like that. So this is the process that we, that we are set to understand. This is the question, this is the aim of this study, to try to understand this process of differentiation from some early progenitor or some non-beating entity to being part of the beating heart. So I hope the question is clear. Okay, so here it is how it goes. They go to the chick embryo, and you need to be quite skillful to do this. They remove the region which is the anterior heart, and they put this explant in a dish. And it sits there, and you wait 24 hours. Of course, you give some serum, you give some food to these cells, and they live, but nothing happens. It's inert, it sits there. Now you do another experiment, and you treat this set of cells with a single morphogen called BMP4, bone morphogen protein. One single molecule you add to this. And then, after 24 hours, it starts beating. Now, this, when they told me the story, so they came to my office and told us the story, and I said, this I don't believe. Why? For me, an embryo is one of the most complex systems you can imagine. I mean, from one cell, you go through some steps, and in a very short time, you get a baby, a fully blown human being. Everything is there. And it's a major miracle. Now, you go into this system in the chicken. You cut out a piece. You put it in a dish. And you put one single morphogen on it, and it starts beating. I mean, that didn't sound something that I would accept. Okay? Because if you cut it out, I was sure that on this road that goes towards the heart, so many things happen that make this progenitor into a muscle cell. So here they say a single morphogen will do the job. So let me show you a movie of that. So they said, well, okay, when I said I don't believe, I said, no problem, come to the lab, and there is a microscope, you can see it. So I went to the lab, and I looked into the microscope, and there was a problem that when I look into a microscope, the only thing I see is my own eye. So they said, no problem, we'll show you a movie. And this is the movie. So it's nothing dramatic, so just watch it. It's a mush of these cells. It's not a single, it's 4 different. You see the beating? It's 4 different explants next to each other. You see that? There it goes again. Now, this is a very interesting phenomenon. This is a macroscopic phenomenon. The number of cells is about 1,000 cells going from one end to the other. And so this is a collective phenomenon over a very large number of cells. They talk to each other. They are coordinated. It's not just a single cell which changed and does something interesting. So how does such a collective phenomenon emerge in response to a single molecule? Okay. So I hope these examples conveyed to you my first point, that the phenomena are immediate. You can look into the microscope if you know how to look into a microscope and see it. And they are very exciting and interesting. If you don't think this is exciting and interesting, then you might as well get out, because this is going to be the most interesting thing you're going to see. Yes?
Is the frequency of the beating directly proportional to the concentration of the
I don't think so. I don't think so. They didn't. I think that once you are above a certain threshold with BMP, then the differentiation process is initiated, and it goes, and once it's differentiated, it is a cardiomyocyte, and it responds like cardiomyocytes respond. Okay, so I was very surprised. So the question is, how does BMP induce beating? And you try to bridge the gap from the molecular level to this macroscopic collective behavior. There's a question back there. Please. I can't hear you. These are the cells that were taken out from the secondary heart field, which in normal conditions would migrate towards the heart and become differentiate into cardiomyocytes. So they cut them out before they reach that destination. If you use some random cell, this will not happen. Okay?
But doesn't that go with your, let's say, prejudice that other things have to have happened? Like you said, that development is a complicated stage by stage something. So in some sense, in the progenitor cells,
something must have already taken place.
Right.
So just hang on a second and come back to me in one slide. So this is the aim, and they approach the question by before I tell you what they did, the question that my student, the one you saw, he's an extremely good student. His basic training was undergraduate as an aerospace engineer, and then he did a master's in applied math, and then he came to me. So he took an engineering approach to the problem. He says, what does it take, just like the Hanahan paper for cancer, what does it take for a cell, for this assembly of cells to beat in a collective way? What has to be there? So there must be a cardiac action potential, and there must be a communication infrastructure. The ions have to flow from one cell to the neighboring cell so that current can go, and that's the way that things are synchronized. And finally, beating is a mechanical thing. And the mechanics are accomplished by a unit used sarcomere, and it has actin, and it has all kinds of myosin-binding proteins, and troponin. All these, there are 20 proteins here whose names you cannot see, that are needed to be there for this sarcomere to work, for the muscle to contract. So these are the three components that have to be in place to see this beating. So then the question that he asked, which of these components is induced by BMP? Okay? And as you said, all are needed, and it's pretty unlikely that everything is assembled in response to one single morphogen. So they did time-dependent gene expression analysis. So you already saw these heat maps, but again, each row corresponds to a gene, each column to a time point, 0, 3 hours, 12 hours, 24 hours. The left side is this experiment done on untreated cells, cells that were not treated with BMP4, and the right side is the time series for cells that were treated with BMP4. And you see here the expression profile, and again, red is high and blue is low expression, and you see it here for about 400 genes. And these genes, so what are you looking for? You are looking for those genes that went up in response to BMP or went down in response to BMP, but did not do this behavior without BMP. Right? So you are looking for things that changed when BMP was added, but did not change or changed much less without BMP, without the morphogen. So then there are 400 genes that pass this filter, and here they are. And what he discovered was that all the electrophysiology-related genes were not affected by BMP. All those genes that have to do with the action potential, that have to do with the ion channels, they were expressed already before BMP was added. But the sarcomere, all those 20 sarcomeric genes, they came up in response to BMP. So it's clear what BMP did. These early progenitors that we had already had some of the machinery in place, and apparently BMP just added one more essential one, the mechanical one. So that is shown on this. This is a typical biology slide. It has 20 figures, no definition of the axis, you cannot read anything. But what you see here, these are the names of those genes, myosin heavy chain. I mentioned myosin on purpose, and tropomyosin 1, type 1, whatever, T type 2. All these are the 20 genes that were represented on the sarcomere as essential for beating, for the mechanical contraction. And what you see in this time is going on the horizontal axis and starts at 0. That's when BMP is added 24 hours later. And in blue, you see the response in the untreated cells, and in purple, the response in the treated cells. All of these went up. So we understand that the way BMP acts is through inducing the components of the mechanical part of this machinery. So that's good. So now we ask what other pathways are affected by this. And we look at the BMP pathway, the pathway of BMP itself. You know, it is a small molecule, and it is bound to its receptor, and the whole cascade happens. And these BAMBI, SMAD, all these are part of the BMP pathway, and indeed they go up in response to BMP as expected. That is not a surprise. But then there is another pathway. There are genes that belong to the FGF pathway, fibroblast growth factor pathway. And all 8 of them go down. Without treatment, they are more or less constant. That's the blue line. I'll move out of the way. And with BMP, that's the purple line, all of these significantly are reduced, the expression of the FGF pathway. So this was a major surprise. Why? Because there was a paper published just a year or 2 before we started working on this, which showed that FGFs are essential for heart formation. They took mice, they knocked out these FGF genes, and the newborn mice didn't have a heart. So FGF is essential for heart formation. What we show here, that in order to get cardiomyocytes, FGF is downregulated. So it's surprising. So there are 2 possible scenarios. Yeah. They were born, and there was no heart, or there was a very small degenerate heart tissue in these newborn mice. That's what I meant. It's not that they were nasty. Okay? So there are 2 possible scenarios. One is that FGF, when it's present, this growth factor, the pathway is activated, and it inhibits differentiation. So when the FGF pathway is on, one of the things it causes is shut down of this process, or arrests this process of differentiation, of expression of the sarcomeric genes. That's one scenario. And then BMP comes and shuts down FGF, so the brake is removed and differentiation can proceed. The other scenario is that BMP somehow, in some way which we don't know how, induces differentiation, the expression of these sarcomeric genes, and independently shuts down FGF. That is, there is no causal relationship between the shutdown of FGF and differentiation, whereas here there is. You understand the difference between the two scenarios? So what we set out to determine or to answer which of the two is correct. So we did a combinatorial experiment. It turns out that you can play with the level of activity of the FGF pathway. You can turn a knob. And the knob is called SU5402. It's a small molecule which, when present in the cell, it binds to the intracellular leg of the FGF receptor and inactivates it. So if you add this ligand or this molecule, it shuts down the FGF signal. So that's the shutdown state for FGF. You can leave things as they are without adding this object. That's the endogenic situation. That's what you saw the movie of. Or you can add ectopic FGF. You can add FGF, just pour it on the thing, okay? And then you will overactivate the FGF pathway. So by this knob, you can take the FGF pathway and take it from a very low level of activity to the endogenic level of activity and to a very high level of activity. Okay? So now there is another knob, which is the BMP. There is a ligand called Noggin which binds to this morphogen. So if you add Noggin, then even the BMP that are in the solution, which have been secreted by the cells, even that BMP is bound and inactivated. So by adding Noggin, you put the BMP pathway at total shutdown. Then you are endogenic, which is don't do a thing. That's the control experiment. Or you can add ectopic BMP. You can pour BMP on the system, which is what we did in the second experiment, and then the BMP pathway is fully activated. So there are three ways, to three operating levels. Here and three. Here 3 times 3 is 9, so altogether you have to 9 experiments. So here are the 9 experiments. On this axis, you see the FGF signal. This is where you have added ectopic SU, so you really shut the FGF down forcefully from the outside. And here, you add the ectopic FGF. So you have activated the FGF pathway at the highest level. And the same for the BMP signal. So these bars represent the level of activity. The orange ones, the level of activity of FGF, and this is the level of activity of BMP, and hard denotes beating. So this is a phase diagram. And you have there is a line which separates two phases, the beating and non-beating phase. And we see that beating is essentially controlled by the FGF signal. When the FGF signal is low, you get beating. When the FGF signal is high, you do not get beating. So this answers the question that we posed, which is that differentiation is apparently governed by the presence or absence of FGF. And what BMP does, it shuts FGF down, removes the brake, and then these sarcomeric genes can go up. So I think we answered the question. This is the correct scenario. The way BMP induces beating is by inhibiting FGF, which in turn removes the brake and causes differentiation. Nice. So here are two main lessons I learned from this study. First, the first one, remember, there was a surprise, because there was this paper which said that FGF is essential for heart formation. So how come that in our study, you needed to knock FGF down in order to get a cardiomyocyte? So the resolution is that FGFs are indeed essential, but they're essential for proliferation. To have a heart, you need to have enough cells that will become a heart. And the growth factor made that. But when there is enough stuff, then you have to stop the cell division and switch to another mode, which is differentiation. So I hope this is clear. And the lesson is that when you have a large-scale manipulation of a complex system, it's very hard to deduce what exactly went on. You all know this experiment about the guy who studied the auditory system of the cricket, that he put the cricket on a table. You heard about Yosemite Newton. And went like this, cricket jump, and the cricket jumped. And then he cut off one of his legs and went again, cricket jump, and it jumped. When he was done, cut off the last leg of the cricket. When cricket jumped, it didn't jump. So he wrote down in his notebook, a cricket without legs cannot hear. So you have a complex system, and you do a very large-scale manipulation, taking FGF out of the system, and there is no heart. And then you conclude that it's essential for heart formation, and indeed it is, but it is essential for one part of the process, but actually interferes with another part of the process. Okay, so this was the first story. No, sorry, there is another lesson. I promised 2 lessons. This is an interesting one also. So this was the question that we posed in the beginning. How do we go from a molecular level of adding BMP to from a macroscopic, phenotypic level of beating and sarcomers? We didn't know. And we ended up identifying how it goes. BMP acts by shutting down FGF. And it was his FGF which actually arrests differentiation. So to me, this looked like a very interesting finding. So you submit the paper, and a review comes back and says, OK, very interesting, but now please tell us the mechanism by which BMP shuts down the FGF and by which FGF arrests differentiation. And when you do these 2 things, then we may consider your paper for publication. So I was kind of surprised by this reaction. And it turns out that in biology, when you solve one problem, you generate 2. And actually, I was even more surprised when I discovered that this is not my discovery. He's an American economist who lived in the, I don't know, end of the 19th and the first part of the 20th century. And he said that actually, the outcome of any serious research can only be to make 2 questions where there was only 1. So even this we didn't discover. But anyway, the reaction of the reviewers was something that taught me a lesson about reviewing process in biology. OK, so this is the end of the first story. If you have any questions, pressing questions, ask, then I'll go into the second story. Wait 1 second, someone is running to you with a microphone.
So don't you find it surprising that just in a macroscopic matter, a single molecule was able to change things so much?
Yeah.
I mean, usually it doesn't happen in physics. You need a finite concentration.
Well, it's not adding OK, I'm sorry. It is a single morphogen. It's not that they added 1 molecule to this pile of cells. They added a finite concentration of the stuff, but it's a single morphogen, a single molecule, not a whole mixture of things that trigger all kinds of other things. Good, I'm sorry. It's a typical case when you use one language and you're misunderstood. I'm glad you pointed it out. Any other question?
What causes the synchronization?
Well, that question is outside the realm of this talk. It has nothing to do with differentiation, because there is an electrical signal which is generated by some pacemaker mechanism. And when it fires, then the ions flow from one cell to another, to another, to another, and the electric signal, which is extremely fast, sort of synchronizes and tells the cells contract. The contraction is a mechanical thing. It's slow. But the passage of ions is extremely fast. Yeah? Yeah, you have, and sometimes And again, it's a very good question, because when you repeat the experiment, sometimes it doesn't work. So in physics, usually you control the initial conditions much better than in biology. And here, in some cases, they succeed. They take the experiment out, put BMP on, wait a day, beats. And another student comes, tries to do it, and it doesn't happen, and the advisor screams, you don't know what you're doing, and you're making a mess. But you have to get enough experience to know when to go in and where to go in, and otherwise you don't see the effect. So not always. It's not a 100% hit rate. Yeah, please, last one.
What part usually differentiates first? The brain?
Sorry?
The brain? Does the brain evolve, differentiate it first, the neurons? Because in some sense you could say that in case the brain plays a part in differentiation of the embryo.
Brain?
First you from the embryo you have the brain developing first, let's say.
Okay.
So then it makes sense because electrical signals are there.
Okay.
So and by its very nature it's useful for coordination.
Okay.
So and the rates will also be faster compared to chemical
Brain
transport.
development.
Yeah.
Yeah.
So
okay.
is it possible,
To do this on brain?
Brain-directed differentiation, does it happen?
Well, this I don't know. I mean, the way, I'm not a developmental biologist. So I'm sure there are people who work on this guy that I collaborated with. He specializes on heart development and heart regeneration. Okay. I'm sure there are labs. I don't think there is one at Weizmann that do brain development.
Okay, because
and the way I do this, I'm not a developmental biologist. These guys walked into my office and said, listen, here is a problem. Okay, it was actually kind of funny. I tell you the story even though we don't have much time. And the guy came in with this lady in tow, and they sat down and he said, okay, I'm Eldad, and I have a problem, and we did some gene expression, and you guys are experts on gene expression data analysis. And we have already done the analysis. We have found genes that go up. We found that genes go down. And she, the student, insisted you know, I'll give you a cup of coffee, and you tell me this story. And we wasted half an hour. You already had invested 15 minutes in it, so what the hell? So then 2 years later, we wrote this paper. So I'm not trying to understand development in general of chicks. But the problem popped up. It looked so interesting that we were sort of dragged into it. So I don't know about brain development. Okay,
may I ask a question? So you're saying that it's kind of...
I'm sorry?
So it is very surprising that one morphogen...
Did this. To me, it was very surprising.
Yeah, but if it is surprising, why did they do these experiments? How did they think about it?
Well,
means that adding this morphogen can... means what is the motivation?
I really don't know. They may have tried. They know that BMP is playing a role in differentiation of cardiomyocytes, and they probably added various things, and this is the one that made things happen. Maybe they had some advanced guess that this is a good one, but they probably tried several, and therefore they hit this one. They wanted to understand the process. Okay? So this problem grew out of a collaboration with the lab of Yossi Yarden, who is a very senior guy who works on these tyrosine kinase receptors. And this is his student. He's an oncologist from Vienna, a medical doctor who did a PhD. And this guy is the same student of mine. Grew up on a kibbutz, which is a farm. So you couldn't imagine more different personalities than these two, and they hit it off amazingly. So again, since they hit it off very well, a collaboration started, and it was running for 3 years, and it's still ongoing, even though both of them have left. So here, the question, there are more people involved, so I'll show them. I'll not go into details what everyone did, but the aim is to do genome-wide measurements of transcription and degradation rates, and to see the dynamics of the process, what kind of dynamics comes out. And okay. So here, the problem is the phenotypic response of cells to stimulus. In a way, it's like before. There is a single thing you add. You add EGF, this growth factor, and the cells respond. There is a phenotypic response. The cells we use are, in this case, so-called MCF10A cell lines. These are human breast epithelial cells. They are not cancer cells, but they have been living in cell lines for a long time. And they are, the response is migratory response. They start to move when you give them the stimulus, which is adding EGF. And then there are two more systems that we looked at, which I'll talk about later. So that's the problem that we look at. And there is a movie that shows the thing. So to understand the movie, you take these cells, you starve them, and you put them in a dish, and they form a monolayer. Now you come and you scratch the monolayer mechanically, and you introduce a wound into this monolayer. You add EGF, that's time 0, and you watch the cells starting to migrate. And that's what you will see on this movie. Oops, no. You should see it on this movie. So this is the scratch, and you start the process, and you see that you see migration. You cannot not see it. The gap closes. And this process, they start moving after a few hours, 10 hours, I think. So this is what we want to understand. And the way we go about it, we look at we do transcription profiling, and we measure the level of expression of various genes as a function of time. And what you see here, this is a gene called vinculin. And time 0 is when we added EGF. And what I plot here is a fold change of expression of vinculin as a function of time. So divide the expression level at 20 minutes by time 0. 40 minutes, 60 minutes, 2 hours, 4 hours, 8 hours. And you see it going, it goes up and then sort of steadies. And the maximal fold change is a bit above 2. And here for TGF-alpha, you see the same kind of plot, and the maximum fold change is around 7. Both of them peak at 2 hours. So there is some interesting dynamics of going up, coming a bit down, and settling down. So the question is, can you do this kind of measurement genome-wide, not just for 2 genes? And you can. And here you see the signal for 440 genes. Again, every row is a gene. Every column is a time point. These time points are that you see here. And the colors are blue for low, red for high. And all these genes were selected because they went through some filter, which was, in this case, it has to go up. And the maximum fold change has to be larger than 2.5, I think we used here. And there are 440 such genes. And we arrange the genes according to the peak of the transcription. So these ones, a small group, peaked at 20 minutes. These peaked at 40, 60, 2 hours, 4 hours, and these peaked at towards the end of the experiment. So they may be still going up, but we stopped the experiment. And what you see here is messenger RNA. Okay? And the 2 genes that are shown explicitly correspond to 2 lines in this heat map. I don't like to ask an audience, do you understand this? Because I never get a response. So do you understand? Is this clear what this heat map represents? Okay. So what do we see here? Well, first of all, as I said, 440 were induced with fold change above 2, and the genes were ordered by peak time. These are called immediate early genes. They peak at less than 1 hour, so they are considered to be primary responders. The EGF, the stimulus, induced some transcriptional activity, and these genes came up as a result of that transcriptional activity. Whereas these genes are designated to a class of delayed ones. They peak after 4 hours, and the common knowledge is, or common wisdom is, that they are probably secondary products. So the first wave, EGF induced some RNA, transcriptional events, which got translated into protein. The time scale there is an hour. And those proteins may have induced these. So everyone measures mRNA, and everyone, when they see something like this, that's what they say, including us in the paper I showed you yesterday. So now I'll give you a very brief 2-minute course on transcription, and we'll take a detailed look at what transcription is all about. So the first step, here is DNA. The black line represents DNA, and this is a gene, and it's made of exons and introns. Each of these vertical blue lines represents a segment of the DNA, which is called an exon. And in between two exons, there is a segment which is called an intron. So the first event is transcription. Polymerase comes and transcribes the entire gene, exons and introns. Okay? And what is generated is called pre-mRNA. While this is going on, a huge machine assembles on the transcribed pre-mRNA and cuts out the introns. This is called splicing. And when you cut out all the introns, what you are left with is a bunch of exons glued together. And this is your mRNA. There is other steps of processing, and then this mRNA is transported out of the nucleus and is translated into protein. The introns are degraded very fast after being cut out. So this is pre-mRNA, which includes the introns, and this is mRNA, which is exons. Then this is translated into proteins, and the time scales are as follows. This splicing is minutes. Pre-mRNA lives very short. It lives a few minutes, and it's spliced. mRNA lives for hours. When it's transported out from the nucleus to the cytoplasm, the ribosomes assemble on it, and they translate it, and within an hour, 2 hours, 5 hours, the mRNA is degraded. Okay. So the question that we ask, can you or can you not, or how to measure pre-mRNA? Everyone who shows you heat maps of expression, they measure the mature mRNA. So can you measure this? Why is that important or interesting? Because pre-mRNA is directly sensing production. How much mRNA you have there is the result of some balance between production and degradation. Pre-mRNA is degraded, turned into mRNA within minutes. So the amount of pre-mRNA that you see now is equal to a very good approximation to whatever was produced 2 minutes ago. So by measuring pre-mRNA, you get a direct handle on transcriptional production of the pre-mRNA, of the transcript. Okay. Yes, you can measure pre-mRNA, and here it is. And this is a thing of beauty and a tremendous surprise. Why? What you see here, first of all, what is this? What we did, we measured for the same 440 genes, the pre-mRNA, and we arranged the genes according to the peak of the pre-mRNA. So if you look at this group of genes, those that peak at 8 hours, within them there are subgroups which we ordered according to the peak of the pre-mRNA. I hope this is clear. We ordered here according to the peak of the mRNA, and all these had their mRNA peak at 8 hours. Within that group of 100 genes or 80 genes, we reorder them now according to the peak time of the pre-mRNA. And this is a huge surprise. Why? Because you see, all these, the mRNA, for all these, the mRNA peaks at 8 hours. And they are produced, some of them are produced after 20 minutes, some are produced at 40 minutes, some are produced at an hour, all over the place. So by measuring the mRNA, you don't have a clue when the transcript was produced. And everyone and his cousin is taking the mRNA data and using it in order to construct networks. This activates that and this activates that. Total, well, I'm not saying nonsense, But you have to look at the actual production, not at the peak of the mRNA, which is a balance between production and degradation. Will in a moment. Okay.
So, in the very early genes,
Yeah.
so later on, the mRNA peak always occurs later than the pre-mRNA peak. But for the very early genes,
Well, yeah, you can see that it doesn't, you cannot have the mRNA peaking before the pre-mRNA.
so if we do see that, it's just noise.
Yeah, I think so. That's right. The interesting stuff you start seeing here and at later times. Okay. So again, there is immediacy of phenomena. My student showed me this graph on my screen, in November 2009. And I was stunned, because here I am, and I am the only one, apparently, who knows that there is a fairly random delay between production and the peak of mRNA. So this is immediacy of phenomena. I mean, you understand, this was done in a lab which is, like, I don't know how many square meters, by one student, and the analysis by one more student. There were no 3,000 guys running around this experiment. So the phenomena are really very, very immediate and very surprising, very interesting. And such basic things you would expect that they know, but they don't. Okay, so another surprise was what we call production overshoot. And we may be to emphasize this aspect. But it was to us very surprising. In some cases, like these two genes that I picked in the first graph, this is the mRNA curve, and this blue line is the mRNA curve of TGF-alpha. And the red curve is the pre-mRNA fold change. To produce a fold change of 7, the pre-mRNA shot up for 15-fold. And here, the production of the pre-mRNA shot up 12-fold to produce a fold change of 12. So this we didn't expect, that there is such a change between the profile of pre-mRNA production and mRNA abundance. There are about 80 genes that do this. And for them, we did some painstaking experiment, not all of them, but many of them, to show that actually this is correct. We did it by RT-PCR, not by microarrays, which are a much denser set of time points. So now answering your question, Matteo, how was this measured? So this wasn't the intention. The whole experiment was not something that we set out to do, even though that's how I present it. We looked at another problem. We looked at the problem of alternative splicing. That was the aim of the experiment, to understand the dynamics of alternative splicing. If you don't know what that is, I said that in the splicing, you glue all the exons together, but not always you glue all of them. Sometimes you delete one, and sometimes you delete more than one, and sometimes so there are all kinds of combinations, and that's the manner in which you can generate 1 million proteins out of 20,000 genes, because there's a combinatorial thing coming in. And so that's what we wanted to study. In order to study that, we used what's called exon arrays. Exon arrays measure the abundance of the mRNA that is associated with every exon. So the student, Amit, who looked at the data, realized that this exon array, just like you expect, it has probes sitting on the exons that measure the exonic abundance or concentration. But lo and behold, there are probes on the introns, which Affymetrix have put in for some reason. About 40% of the probes, there are 1.5 million probes on this chip, 40% of them sit on introns. And the intronic signal is usually used in order to calibrate. And Amit says, this is basically treated as garbage and thrown out, so let's take a look at this garbage. And then he thought about it, and he is the one who had this idea that the reading of these probes actually reflects the pre-mRNA amount. So this gives you a way of measuring genome-wide pre-mRNA abundance. Okay? So the exonic probes, the exons are present in the pre-mRNA, in the mRNA. So the exonic probes actually measure the sum of pre-mRNA plus mRNA. But the pre-mRNA is about 1% usually of the mRNA, usually. And therefore, we can say that I measure exons, I measure mRNA. I measure introns, I measure pre-mRNA. Okay? So did I answer your question? These are the equations that govern the process. In the books, what you will find is a single equation. M is the concentration of mRNA, and P is the concentration of pre-mRNA. In the books, you find only DMDT, and it usually says DMDT is beta, production of messenger RNA, minus alpha times M. This is degradation. But you have to use two equations and write one equation for pre-mRNA. This is what is being produced, actually, and degraded. Its degradation is due to splicing, and whatever is disappearing here reappears here. So the source term for the mRNA is the pre-mRNA that was spliced out. Wait a minute. And then it's degraded. So this process is shown here as a caricature of these equations, that here is the production of this entire object. Splicing generates pre-mRNA, mRNA, and mRNA is then degraded. And that's what's described by these equations. Question?
You had regulated degradation of the mature mRNA, but the variation of the delays between the pre-mRNA peak and mRNA peak suggests that even the alpha-1 might be a time-dependent effect.
Well, in fact, it's not. We measured it directly. It's about, the time constant is minutes, 2 minutes, 3 minutes, and the variation is pretty small. Yeah. I'm sorry, if? What happens in? Oh, in terms of what?
Is it due to difference in the temporal pattern?
Here is the, okay, so the question was about what, how this gets modified in cancer.
Yeah.
Look, the main, I don't know. And we did not do these experiments on cancer cells. What I can say is that probably people believe that regulation or deregulation happens on this level of production. Genes that are supposed to be expressed are shut down, all the tumor suppressors, and those that should be silent are activated. These are oncogenes. And I believe that is largely correct, but as you will see if you wait just 2 more transparencies, we discovered that degradation is also varying with time in a non-trivial manner. So I will be very surprised if someone, and there are people working on mRNA degradation, but for everyone who works on degradation, you have 100 who work on production, okay, including the difference between normal and cancerous condition. So I would be very, very surprised if the malignant transition does not take advantage of manipulating the degradation as well. I'm sure. No, I don't know whether people really took a careful look at that. My guess is no. So we measure P and M as a function of time, and then use these equations to infer the time-dependent production and time-dependent degradation profiles. So I'm really going a bit fast here, because to do this, you need to take data at a rather dense set of time points. You cannot do this with microarrays because they are expensive and they are not that accurate. So we did PCR on a selected set of genes, and then we were able to actually measure directly the pre-mRNA by looking at the exon-intron junction. Okay? That, you have that object only in the pre-mRNA. The intron itself is floating around a bit also after it has been spliced out, but the junction dies as soon as splicing took place. So this is the result of this experiment for 4 genes. It's pretty amazing. I mean, I picked these because they are pretty amazing. You see that the red line, which you can see the colors, the red line it has the red dots on it. It's the measured pre-mRNA. The blue dots are the measured mRNA. And the green curve, which really is very close to the red line, is the production that we inferred. And the blue one is the mRNA concentration, and this gold one below is the degradation profile as a function of time. So let me magnify that for you, for one particular case. This is degradation as a function of time. It has a non-trivial behavior. This gene, the mRNA, goes up 150-fold. In order to jack up the level of mRNA 150-fold, the system generates a peak of production which is 600, but that's not enough. It actually, what it does, in order to get mRNA up that high, it shuts down degradation. So the gold line is the degradation profile, fold change with respect to its value at time 0. And this goes actually to 0. So the degradation is really shut down for some time to get the mRNA level that high up. It's pretty amazing. So why is the system doing this? Overshoot and all this playing and stabilizing. The aim is to get the right amount of mRNA and eventually protein there at the right time. It has to get there fast. So in a way, I'm trying to explain this strategy of the production peak, of this narrow and very high peak. So when you drive a car and you are just about you want to get on one of these highways, and there is a flow of traffic. So you want to get from the speed that you are at when you are on the ramp to the cruising speed on the highway. Let's say you are driving at 20 kilometers per hour and you have to get to 100 kilometers per hour to merge. So there is one way is to put your foot on the accelerator right at the level of cruising at 100 kilometers per hour. Cruising means once I'm already there, this is where I keep my foot to stay there. So the car will climb up to 100 kilometers. It will take a while. And there are people who drive just like that. They are not very popular with the other drivers. What you normally do instead, the Americans call it pedal to the metal. You slam down on the accelerator all the way, give a big burst for a limited time, which takes you up to that speed, and then you raise your foot to the cruising level. That's what cells seem to be doing. All the way, slam down, and then go. So someone asked me at one of the dinners or lunches here about energy. Do cells try to optimize the amount of energy that they spend? This is not optimal in terms of energy expenditures. But there is enough energy, apparently. There is enough food. It's much more important for the cell to have the right component there, ready at the right time when it wants to start to move. Because moving is what it has in its mind. Okay, that's the strategy. So these few slides held us back with publication for a year. Because the guys were aiming high, and they said, well, we have to show that this is not only the MCF10A cells in Yossi Yarden's lab who do this. We have to look at, in physics, it's called universality. You have to show that everything does it. Now, universality in biology is to show that at least one more system does it. So we were, since I'm a physicist, we took a big step and said 2 systems do it. So we looked at very different systems. These are murine dendritic cells from bone marrow of mice, which respond to stimulation by an agent called LPS. And they mature these cells, and this is then produces inflammation and maturation. And we found again genes whose response is very much like what we saw in MCF10A. The mRNA concentration goes up by a factor of 2, and there is an overshoot to factor of 9. Here is similar situation. And then the last system was human embryonic stem cells, which differentiate into neural progenitors in response to retinoic acid. And there again, we saw some similar kind of behavior. So we have experiments from the first system, the mammalian, the cell lines, and then from mice and from human embryonic stem cells, and we see this behavior. Yeah.
I'm trying to understand the mechanism behind this. In your curves, the mRNA degradation, the uptake of degradation happens at different time points for different mRNAs.
Yeah.
So the sequence dependence of that would seem to suggest that the degradation mechanism has all kinds of different factors which are very sequential.
Yes, yes. The degradation happens by binding of proteins which are responsible for degrading mRNA onto particular positions on the mRNA.
But you would have to have as many proteins as the different mRNAs you're trying to degrade in a sequence-specific manner.
Yeah, there must be something like that. A protein can bind, initiate degradation, unbind, bind to another mRNA, initiate degradation, and so on. So I don't know how, it's not one-to-one. We looked at length dependence. I don't remember. There is some length dependence. Okay. So this is the summary. I'll skip that because I took a lot of time. And as you see, this is going back to my first slide. The last point I added here, that submitted papers are not reviewed. In some journals, this was not reviewed, to my surprise. And the reviews are often completely idiotic. We got back a review from Cell, a one-line review after a month, saying all this was seen in yeast. It so happens that yeast has 6,000 genes and only 200 introns. So yeast doesn't have introns. So none of this could have been seen in yeast, at least Cerevisiae. The other ones, that are some that have more introns. But none of this was seen in yeast that we saw. Okay. Breast cancer. How am I doing with time? I'm still okay? So the breast cancer. So again, these are the people whose picture you saw yesterday. And this is the paper in Nature by Van Tvier et al., who claimed that they found 70 genes whose expression levels can predict whether an early discovery breast tumor is aggressive or not. Okay, so this is me. And this you saw yesterday, but let me just quickly go through it. The death rate from breast cancer is about 30 per 100,000 in the population. Very high rate of incidence. We look at early discovery tumors. The standard treatment is resection, radiotherapy, and then if there is overexpression of estrogen receptor or progesterone, then there is hormonal therapy. And if there is overexpression of HER2, then they give the patient Herceptin. And the big question is chemotherapy, systemic, general chemotherapy, yes or no? And here the answer is taken using these criteria that are clinical. And these criteria generate severe overtreatment of these women. About 80% of these women were getting chemotherapy under these protocols, whereas maybe 20% really needed it. This is what I just told you. So I'll skip this slide. So again, the problem is very, very simple to state. You don't have to be an expert. You don't have to understand that this is a serious problem. The only thing I didn't tell you that you have to know, but most people know, is that chemotherapy is bad. It has adverse side effects that really make you sick. And even sometimes, if they don't make you sick, they have all kinds of psychological side. People don't like when their hair falls out. They don't die of it. But it's so, and on top of it, it's extremely expensive. When I started working on this in I don't remember, 2004, I was for a short sabbatical in Institut Curie in Paris, and the guy who introduced me to this subject told me that at that time, France was spending €1 billion a year on this kind of systemic chemotherapy, which was 80% of that was not really needed. So it's a problem, a real problem on many scales, and it's very easy to explain. And it's important. And so let's go to the next one, and this is the experiment I told you about, the paper by the Amsterdam group who produced this signature, and the Rotterdam group, and there was a very small overlap of 3 genes. Here I put 2, which is a mistake, 3 genes between the 2 experiments. And the question was why, and this was the standard answer given. Don't worry, it's different platforms, different women, different types of analysis. And we have shown that this is not the case. And again, the way I'm repeating this because this lecture was supposed to happen also in physics, and maybe I thought there are some people here who weren't here yesterday, so I'm repeating this. So how did we show that this is not the case? We focused on one experiment and essentially repeated it in silico, computationally. And the way we repeated it, I explained yesterday, maybe I think I'll explain again, and I'll try to answer the questions. Why 70 genes? Why these 70 genes? And then these 3 points I did not do yesterday. So this is where you should wake up. So why these 70 genes? As I said, they generated a training set by randomly pulling out 78 patients from the pool of 97 that they had. They used them in order to calculate the correlation of every gene with outcome. For these patients, they knew the outcome. They knew whether there is or isn't recurrence or metastasis after 5 years. If there is not, it's good outcome. If there is, it's bad outcome. This was their outcome label. And then they calculated the correlation of each and every gene expression profile over the 78 patients with the outcome profile over the 78 patients. And then they ranked the genes according to this correlation. And there were 5,000 genes that were expressed. They were ranked, and then they used machine learning. They used a very simple classifier to separate the good from bad outcome. They started out with using the top 15 genes on the list, and they measured the error of classification by leave one out cross-validation. And then they added genes, and as they added genes, they discovered that the error goes down. This is standard procedure in machine learning. And it went down until it started to go up and to fluctuate. And then they said, this is the best number of genes, 70, and then I said why the number 70 came out. Probably because they had 78 samples in their training set, which you're trying to fit. Had they had 700 patients in the training set, my guess is that the 70 would have turned into maybe 150 or 200 or 250. So there was nothing sacred about the number 70, and they have a chip where they print these 70 genes. It's called, the company is called MammaPrint, and this device is sold for $3,000. Okay, and the 70 genes there could have been much more. So, having gone through this yesterday, we got this answer that number 70 is not based on any scientific insight. And then why these 70? Where it's clear that these are the 70 top ranked ones. And then we asked, how reproducible are these genes? And the way we asked that question, we generated another training set of 78 patients, just like theirs, and repeated the whole procedure and got 70 top-ranked genes on the basis of this new training set. It's not entirely new. There is a significant overlap because they were drawn out of 97 patients. And what we found is the 2 lists of top 70 that now are drawn from the same patients. They have an overlap of 12. This is wrong. I missed the one. Of 12 genes. So it's more than before. Before it was 3. But it's not much more. Before, I mean between Amsterdam and Rotterdam, the maximum overlap you could get there is 50, because there were only 50 genes that appeared on the two lists and on the chips. Some of the genes of one group did not appear at all on the chip of the other group, so those couldn't appear. So the maximum you could get was around 55, and they got 3, so it's about 6%. Here the maximum is 70, because we use the same chip. So the maximum is, so they got 12 out of 70, so I don't know, what is it, 15, 20%. But still, it's a very small overlap. So clearly, the small number of overlapping genes is not due to the fact that they use different platforms or different patients or different whatever. It's some inherent instability, yes.
What was the overlap between your samples and their samples?
I cannot speak up.
What was the overlap between your samples? You have taken 78 samples and they have taken 78 samples, right?
There are 97 samples altogether.
For their studies, for training sake, they have taken 78 samples.
They took out of the 97 randomly 78. And I went to the same 97 and randomly took out 78.
Yes, right. So what was the overlap between your 78 samples and their 78?
What is the overlap? Well, the maximum overlap is 75 or 76% out of 97. So the maximum overlap is what? 60? No, not the typical overlap. The maximum is 78. I could have picked the same 78, but the chance for that is very small. Okay? So the typical overlap will be like 60. And it was around 60. Okay? It could fluctuate a bit, but by 5 up or 5 down, not more. So it's not the same patients, but there is a significant overlap. Yeah.
I guess I don't understand what the original study did, because they knew the outcomes of all 97. The 78 out of 97 is a random subset.
Yes.
But whenever somebody does this for machine learning, you obviously repeat this procedure 10,000 times to get some sort of estimate of the performance of your classifier.
Well, okay. I don't know. They published one. They didn't report on repeating it. Okay? Repeating it is tricky. Do you understand why? Because if you repeat it, you already exposed the test set into the thing. So in principle, in these studies, you have to do this jackknife separation of training and test, and the test should be put away and not touched until the very end of the procedure. So in principle, what they could have done is take a training set, okay, take 97 samples. So take 60, and that is my training set, okay, and add 10 more, so you have 70, and then pull again 60 out of the 70, and again 60 out of the 70, and keep the remaining. Okay. Let's say you want to do it properly. So you want to take the test set and not touch it until the very end.
But, so then
So they should.
I don't understand.
Yeah.
Did that happen because of some issues with patients?
No issues at all. It was done in a sloppy way. I mean, that's all. I mean, that was done in a sloppy way. You see, they want to use the patients. They have a small number of patients. They want to use as many for training as they can and still have enough for test sets so that the result they get on the test set has some statistical meaning.
Do it again with a different random sample, and you do it again and again and again to get some estimate of how badly you're performing. That's the standard procedure.
Well, apparently not. Apparently not. It wasn't done. And we were not the only ones. The same year that we wrote this paper, these people from what is called this big hospital outside north of Paris or south of Paris, Gustave Roussy. So they also noticed this, and they also made the same point. They said, look, you have to do it again and again, just like Mukund says, and then you get a distribution of the success rates. They didn't look at the overlap that much as at the success rate, and they looked at a large number of studies, and they showed that the success rate that was reported in all of these studies had a probability of being observed of 10 to the minus 5, okay? Which means something, okay? The Van Tweer's probability, they made the 5%, barely, okay, barely. So, as I said yesterday, the reason is that the number of samples is too small, fluctuations of the correlation are too large. The difference in correlation between these top contenders is much smaller than the fluctuation. So you rank objects by some property whose fluctuation is much larger than the difference between these guys. And therefore, no wonder that when you redo the experiment, everything goes banana, it goes haywire. Okay? And in a calculation that I showed you yesterday, we have actually shown that you need 2,400, 2,200 samples so that two such groups that you generate, random 78 or random whatever, will have an overlap, typical overlap of 50%. This doesn't mean that you need 2,000 samples to do this. It means that this is not the way to do this. Okay? This is the wrong way to approach the problem, in my opinion. Okay, so there was no good reason to get these 70 genes. It could either be basically a set of random genes, but then people come back at us and say, well, who cares? Perhaps these genes give the best prognosis somehow by some miracle, and then why do I care that there are other sets of 70 which you could select? These are somehow the best. So are they? So what we did, we took their ranking. This is their ranking from their paper of the top 1,000 ranked genes. And we took their classifier, the top 70, and we plot here the survival. Okay, we used their classifier to separate the patients into good outcome and bad outcome. All the patients, including the training set. And we plot the survival as a function of time. And you can see that it's a good classifier. You get a good separation. There is a p-value of 10 to the minus 7 to get this kind of separation on random data. So it's a nice classifier. But then you take the second group of 70, build a classifier from them, and you get as good a separation as before. And the third one, and the next, and the next, and the next. The next is not that good, so we skip it and we take the next one. And so on. So in principle, any group of 70 out of these top 1,000 genes gives you as good a separation to good and bad outcome as the top. And therefore, there is nothing special about this group of top 70. They have the same prognostic value, and therefore there is no reason to put these 70 and not some other group of 70.
Sorry.
And then they say, well, maybe you are right, but who cares? If it works so well, use it, and you'll save so many women from chemotherapy. Right? So how well does it really work? Okay, so this is a success rate that was reported by the first Nature paper, which was based on basically 20 patients in the test set. And then you look at another study that came out sometime later, and there the success rate is 50%. And the study that came out later, this is the biggest scandal of all. There is an EU-funded effort, $20 million or something over so many years, of validating this nonsense. Okay? And the paper they published, which had about 300 patients, the success rate was 42 or 43%. So being a physicist, when you see a set of such numbers, you plot them as a function of the number of samples, right? And you see that if you would keep adding samples, chances are that pretty soon you would reach this level, which is the level produced of success rate reported by a software which is freely available based on clinical indicators used by doctors. So does it work? Well, it works, but but not better than classical. So then there was another issue that came up. And this was the one that really got me annoyed enough to keep working on this. Because they were interviewed and they wrote a review about this. And they said, well, you know, I remember the citation in Nature Cancer Reviews of the Amsterdam group saying, well, some claim that there is no overlap between gene lists. Some being me, some et al. And we shouldn't worry about that because the different gene lists probably do represent the same biological processes. Okay, so you remember I told you yesterday about the apoptotic network. It had 88 genes associated with it. So maybe the Amsterdam people caught 2 of them, and the Rotterdam people caught 2 other. So the overlap between the genes would be 0, but they do capture the same process. So they do capture the same biology even though they do not have the same list. And there were some papers written who actually claimed this. So I went in with a student, and we actually looked at this problem, and with a careful statistical analysis, we went through all these papers that claimed to have this overlap of biological processes. The mistakes were amazing. Like, if you know how p-values are calculated, okay, you can ask, what's the probability? So you do an experiment. Well, I will not go into it. It's not worth wasting your time. And so the answer is that there was one biological process that was actually overrepresented in both the Amsterdam and the Rotterdam signatures. It's cell proliferation, which is very nice. It's a sanity check because, I mean, cancer cells, as you heard in the first lecture, that's what they do. They divide. And as for using that for prognosis, the mitotic index, which is defined, it was used 50 and 60 years ago. When people have a cancer, they take a slide, they look at the slide. A slide has 500,000 cells on it, and they count how many are undergoing mitosis right now. That is the mitotic index, and it is a measure of proliferation. And it was used as a prognostic indicator 50 years ago when it didn't have all these molecular methods. So very nice that proliferation came up, but I mean, it would be very nasty if it didn't. And we didn't gain anything new. So, to summarize this story, we have this book. You know, this is a book from the Bible, the Old Testament it's called, and it's a very good book. I recommend it to read. It was apparently claimed to be written by King Solomon, and this is what it says, and since your Hebrew is probably not as good as it should be, I provide an English translation. What has been will be again, what has been done will be done again, and there is nothing new under the sun. Nothing new has been learned, nothing new, nothing significant has been accomplished by any of these fancy signatures. Okay, so that summarizes this part of the talk. And this is an example of what I wrote about physics, that there is an objective truth. Wrong claims either go unnoticed or have very little short lifetime. And in fact, in biology, leading journals publish many wrong papers that linger on. They have a lifetime of 5 to 10 years. Amazing. And it's even more amazing that when such a obviously wrong paper is published in Physical Review Letters, when I used to publish there, if I publish some nonsense, someone writes a comment. And there is no way that the editor will send the comment back to the guy who submitted it and say, this is of no interest. You published a paper on a subject, meaning that it's interesting. If someone shows that that paper is wrong, that cannot be not interesting, by definition, basically. And in physics, as I said, submitted papers are reviewed, whereas in biology, in most leading journals, they are sent back by some robot. And the reviews are sometimes really amazingly stupid. Amazingly. Sometimes they're extremely elaborate, and it's clear that the guy spent a week reading your paper and thinking about it. But sometimes you get back 2 or 3 lines of utter, total, absolute nonsense. Okay?
Excuse me.
Yeah.
There are 2 questions. One is in relation to, if you look at many things as modulating the electromagnetic absorbances to fields.
I'm sorry, I really can't
Like,
understand you. Maybe we can wait with the questions to the end, because I really want to get this done. This will be I apologize. It will take me 5 more minutes, and we are more or less at the end of the lecture, so I really don't want to spend a lot of time figure out how that I give you the right answer. I'll let you get at me at the end. So this is what I told you yesterday about pathway-based analysis, which I think is a hope. And this is the last story, which is part of it I told you. Here, this guy is a physician, and he does research. He is also the PhD. And this is his student. She was, at that time, my student, and she had a strange trajectory. She was accepted to medical school, and then she came to do a master's with me, and then she decided to go back to medical school. And went to medical school and found it totally boring. And she is a computer scientist by training, and she was so bored that she asked them to let her do a PhD in parallel with the medical training. So there are these PhD, MD programs, and then they do 1 year of this, and 1 year of that, and 1 year of this, and 1 year of that. And she said, no, I want to do it at the same time. And there was a big discussion. If someone is interested, I'll tell you it's an interesting story. She got what she wanted, which she usually does, and now she's a psychiatrist. So, but I grab her, I don't let her go, and I force her to come at least once every 2 weeks to Weizmann. She has a desk, and we just submitted a paper on schizophrenia. So anyway, so the observation here is that children with Down syndrome have a 20-fold higher risk of getting this acute lymphoblastic leukemia than non-Down syndrome kids. Down syndrome is trisomy of chromosome 21. And he's very keen on figuring out why. So I already tell you, we don't know. I mean, we haven't answered this question. But this is the aim, the grand aim of the research. You can see here, this is chromosome 21. There are 3 dots. There are 3 copies of chromosome 21. And it was known, it wasn't discovered by us, it was known that this receptor, this cytokine receptor-like factor 2, this is a receptor of a growth signal. That its overexpression is involved in this type of leukemia. It has been known already for several years by this group. And indeed it is, and what we found a reason for this overexpression by looking at the gene. We saw that this section, I showed you this when I talked about gene amplification, to remind you, if you remember. So this is deleted, this whole section of the genome, which places the CRLF2 gene under the control of the promoter of a gene which is very highly expressed. So this is the mechanism by which you generate the overexpression of this particular receptor. And this receptor, and you can see that here, that if you look at the expression levels of this gene and of that gene, when this one is gone, which is this blue, that's when you see this one being high up. Hope you're still awake enough to understand that when the gene is deleted, you will not see it in the expression profile. Okay. And this happens much more in Down syndrome leukemia than in other leukemia of other children. Now, here is another observation. So first of all, this is the pathway to which this receptor sends its signal, and there is a kinase, JAK, which is involved in that pathway. So both of them, if both of them are hit then there will be a very high level of activity of this growth pathway. And that you can see here. These are the cases. This is an experiment in cell lines. This is the case when the receptor is overexpressed and this kinase is mutated. When both events, mutation of the kinase and overexpression of the receptor, occur, you get a tremendous boost. This is the number of cells, a function of time. So production is much faster here than in any of the other cases. And now here is another observation. If this circle represents the Down syndrome leukemic children, the white circle represents those that have overexpression of this gene. About 60% of these kids have overexpression of that receptor. And about 20% of the kids have JAK mutation. The interesting point is that all those who have the JAK mutation have overexpression of the receptor. So there is no green spot here. Is that clear? So this bothered me. I mean, but it really did. I lost sleep over this. And I said, well, if you see this only on this background, it must be that CRLF2 overexpression somehow causes JAK2 mutation. And I really couldn't figure this out. There is overexpression of a receptor due to some deletion which happens on one chromosome there, and there is a mutation in a kinase on the chromosome there. How can possibly this overexpression cause the mutation? And then at some point when I saw Shai, the clinician who collaborated with me on this, and told him that this is bothering me, he said, no, there is no causal relationship at all. It is selection. There is a certain low probability of this JAK mutation when it happens on the background of amplified receptor. Then there is such tremendous proliferative advantage that it takes over. Okay? So this very small probability gets amplified. So now if you look at the leukemic kids who have this receptor overexpression, you will get a JAK mutation. That cell, that one mutated cell, will take over the entire population. Normally, some of them will die. You wouldn't see them. So in this case, it is just pure selection. No causality. And this is the last point I had in my slide. Physicists are trained to look for causative relationship. We live in a world which has causality built in. Whenever we see something, two effects that are somehow related, we ask, how does this one cause that one? And biology is centered on selection. And I think if I have to point to the one huge hole, I think physicists' education is excellent to do just about anything. And one huge hole in our education is that as physicists usually don't learn at all about evolution, which is a big mistake. The fact that we don't know molecular biology is okay. We read the book. But this selection and in evolution is such a deep concept. It's the same way that we are trained to immediately look for causal relationship, a biologist immediately looks for this. So this is something that I try to convince people who talk about curricula of schools that allow physicists to go into biology fast. Don't bother with all kinds of chemistry. They can learn that if they want. But make sure that they learn statistics and make sure that they learn evolution and selection. So this is a big hole in our education. The last point, the biggest upside, the talk is about upside, downsides, and the unexpected side. When I got into this field, it was about 12 years ago. And these are the people I collaborated with. Now, these are PIs in yellow with whom I wrote papers. These are not collaborators that we had tea together. Okay? It's a symbiotic relationship to help them get further understanding and insight to their data. But the number is amazing. I mean, when I gave this talk to the biologist, I said, I'll make a list. I pulled out my publications, and I made a list of the PIs. It's not every paper has 10 names on it. I mean, I don't put the 10 guys here, just the one PI. And it's incredible how collaborative they are. And these are the students that were with me throughout the years. Only those that are in bold are still in the group. The group has shrunk a lot, but at any given time, there were like 10, at the maximum was 20. It was a madhouse, going down again. So this sort of gives you a summary of the biggest surprise and really joy that I had when I moved into this field. My one single major discovery when I became a biologist, sort of, and I started to take students in biology, was to discover that half of mankind is womankind. In 20 years of physics, I had one master student who was female. And after I moved here, people in the physics department were bad-mouthing me and saying, oh, that guy, he takes only women. So this is the summary of my talk, and thanks very much. I don't think there is any more. And again, I overran by 5 minutes. Okay, thank you.
Selection and causality that you talked about need not be mutually exclusive.
Of course not.
Take drug resistance, for example, mutations that come due to exposure to drugs. You know, there are evidences now that there's increased rate of recombination and mutation. So it's causal to some extent, although the linkages may not be understood.
I'm not saying that causality doesn't exist in biology. And we showed that BMP4 causes the shutdown of FGF. But the physicist's gut reaction, when you observe any such dependency, okay, is to look for causal relationships. It's not in our DNA selection. Whereas a biologist immediately just looks at it and says, of course. That's what I was saying. Okay, so you had a question, and which I promised to get to. So, oh, you want to do it while we eat? That's fine. Let's send everyone for lunch. Okay, thank you.